Hello, everyone. My name is Abdullah Shihara. I'm a first year PhD student in Auburn University, and I'm very excited today to talk to you about this research that we recently did. So it is titled as the subserial localization of soybean vein necrosis virus proteins in plant cells reveals nucleolocalization in the nucleocapsid. So as a background, soybean is a very important crop in Alabama. It is the fifth most important planted crop in our state. In 2022, we produced 870 million pounds of soybean, which resulted in a value of $206 million, which is a lot of money. But unfortunately, soybean is being targeted by a virus that is called soybean vein necrosis virus. And this virus was first discovered in 2008 in Tennessee, and in Alabama, it was first discovered in 2013. This virus is causing symptoms of necrosis on the infected leaves that you can see between the leaves or between the veins of the infected leaves. That's why it is called vein necrosis virus. And this virus have a single-stranded RNA genome, and it belongs to an order that is called orthotospovirus. So this virus is composed of three main segments, as you can see here on the figure. We have three main segments, the small segment and the medium segment, and then the large segment. On the small segment, we have two open reading frames. The first one is the NSS protein or the silencin protein. And then the second one is the nucleocapsid protein or the N protein. On the M segment, we have also two open reading frames. The first one is the movement protein or the NSM. And then the second one is the glycoproteins or the GN and GC proteins. Finally, the large segment has the RDRB polymerase protein. Something interesting to know about this virus that other studies that have done phylogenery to show the relationship between orthotospovirus, all of them, they shown that SVNV is distinctly different on the phylogenery from other orthotospovirus. And that was actually very interesting. So I based my hypothesis based on that. So I said, since SVNV is distinctly different on the phylogenery, therefore the protein localization of SVNV will be different from other characterized orthotospovirus. And these four viruses are the one that well characterized, which means that they have a localization for their proteins that we know about it and we know how they function. But we did not know any idea about SVNV proteins, and there was no study before that have shown this before. So this is the first study that ever looked at the localization of this virus, which is very important to know about this virus and how it functions in a cell and how can we manage it. So I used the reference genome, which is the Tennessee reference genome, to amplify the 5 open reading frames, including NSS, N, NSM, GN, and GC. And then I cloned them into the p-enter the topo vector, and then I send them for sequencing to make sure the sequence is good. After that, I fused them to the fluorescent protein GFB in the p-side vector, and I did that in both C-terminal and N-terminal of GFB, which helped me to confirm the results. After that, I transformed this fluorescent construct into the agrobacterium, and I used agrobacterium infiltration in Nicotiana benthomiana plant as a plant model so I can visualize the proteins. Finally, I used the microscopy, which is the confocal microscope, to visualize the proteins. As a result, so we see here the first column shows us that the proteins of SVNV being fused with GFB, and then the second column refers to the transgenic plant that I use, which is called RFBH2B, meaning that the nucleus of this plant fluorescent as red. And then the last column is an overlay between GFB and RFB. And as you can see here in the first results, we have the N protein and NSS protein. They both localize into the cell periphery and into the nucleus of the cell, as you can see here. For the GN and GC proteins, consistent with their domains because they have transmembrane domains and single peptide domains in their sequence, they localize into the cell membrane and also around the membrane of the nucleus. Interestingly, for the movement protein or NSM protein that you cannot see here, obviously, that's why we have a different figure for this protein. Because every time I tried localizing this virus or this protein, it was not fluorescent or it was not expressing, as you can see here, it was always black. And I did that more than 10 different times. But actually what we saw was yellowing symptoms on the infiltrated plants and cell death. The cell death for us appeared as the cells were not in a good shape and we could not determine if this is cell death or not. So we wanted to confirm it. So we used the Cytox stain that is called Cytox blue stain. And basically it's a very easy way to test the viability of the cells. Basically, if you see the cells inside if you see the stain inside the cell, as you can see here, it means that the cells are dead because the stain were able to penetrate their cell wall and go inside of them, which means that they're dead. I used a negative control with the GFBN, which is this one here on top, which did not kill the cells, and I used it as negative control. And as you can see, the stain was just outside the cells. So in conclusion, SBNV was actually different, and it matched our hypothesis that the protein localization of this virus was different from other characterized orthotospovirus. The two big main discoveries is that the nucleocapsid, which is the end, localized into the nucleus of the cell, which has never been shown by any other orthotospovirus. But for NSS protein localizing in the nucleus, it actually has been shown by another virus that is called capsule chlorosis virus, found in this clade here. In the NSM protein that causes cell death, it was very interesting, and this could actually answer two questions for us. So the virus caused symptoms of necrosis, right? As you can see here. So since SBNV NSM causing cell death, which is basically necrosis, so maybe NSM protein is the one causing the necrosis. And also, other literature have said that this virus cannot move systemically on its own in the plant. And of course, NSM is moving the virus in the cell. So if the NSM is toxic and it cannot move the virus, so maybe that's why this virus cannot move systemically on its own, because of the toxicity or the lethality of NSM. So for the future research, we're actually want to determine the protein-protein interaction using the biopsy technique to help us understand why the virus have nuclear localization for its N and for its NSS. And also we have sequenced the virus from Alabama, and we found a lot of mutations in the virus compared to the one that were localized. And we are also interested into localizing these mutated sequences to see if the mutations in their sequence have impacted the localization or not. So that's it for me, and thank you so much for watching. And please, if you have any question, you can email us, and thank you so much for listening.